Artificial insemination is a technique in which sperm are collected from the male, processed, stored and artificially introduced into the female reproductive tract at proper time for purpose of conception. AI is most common method of breeding intensively kept domestic livestock, such as dairy cattle, pigs and turkeys. AI is increasing in horses, beef cattle and sheep, and has been reported in other domestic species such as dogs, goats, deer and buffalo. It has also been used occasionally in conservation breeding of rare or endangered species, for example, primates, elephants and wild felids. Artificial insemination as a means of livestock improvement are now accepted and utilized worldwide. This is possible because a few highly selected males produce enough spermatozoa to inseminate thousands of females per year, whereas only relatively few progeny per selected female can be produced per year even by embryo transfer. The increased use of outstanding proven sires to enhance production potentials, control genital diseases transmitted through natural service which aid in animal improvement results from the expanding use of artificial insemination. Lotsaros Polanzini, 1780, reported first successful use of AI. He started experiment with dog. When one bitch manifested the signs of heat, he used semen at body temperature to inseminate the bitch. 62 days later she gave birth to 3 pups. He is also called father of modern artificial insemination, semen collection. Semen collection refers to the process of obtaining semen from males with the use of various methods, for the purposes of artificial insemination, or medical study, usually in fertility clinics. Semen can be collected via masturbation, example, from stallions and canids, prostate massage, artificial vagina, penile vibratory stimulation, vibroejaculation, and electroejaculation. Semen can be collected from endangered species for cryopreservation of genetic resources. Macroscopic evaluation. The semen should be transferred to a water bath maintained at 35 ± 1 degree Celsius. Visual evaluation for volume, color, consistency, density, odor and observation for presence of foreign material, blood, pus cells, dung, hair, etc., shall be made and recorded. If dung or hair is found in the semen, filtration with special semen filter is done. Microscopic evaluation. Microscopic evaluation is done using a simple or phase contrast microscope for mass activity, wave motion, and individual motility. Determination of concentration is done with a hemocetometer or a calibrated photometer. At this point, if required, smears can be made for morphological studies and live-dead count. Nigrosine stain is recommended. Buffered nigrosine solution is mixed with a drop of semen and smeared on a glass slide for morphological examination. It should be dried and examined under oil immersion. Automated computerized machines for recording motility and concentration and calculating the required extensions are now frequently used in AI centers that can afford them. Various biochemical tests used for assess the sperm fertility. 1. Methylene blue reduction test. 2. Cold shock resistance test. 3. Zone-free hamster egg penetration test. 4. Sperm mucus penetration test. 5. Hypoosmotic swelling test. 6. Computer-assisted semen evaluation. 7. Fructolysis index. 8. Oxygen utilization test. 9. Milovanov's resistance test. 10. Hyaluronidase content. Dilution of semen. The first major improvement in the AI procedure initiated in the United States was the development of a yolk phosphate semen extender. Phillips and Lardy, 1940, improved the media by buffering the egg yolk with sodium citrate. Extender preserves fertilizing capacity of the spermatozoa for a long period and increases the volume and thereby services to large number of females. Diluter provide viability and fertility of spermatozoa for prolonged period. Ideal semen diluter should be isotonic with the seminal plasma, have a pH of 6.6 to 6.8 with high buffering capacity, contain lipoproteins and lecithin, minerals in adequate quality and substances used for aerobic as well as anaerobic metabolism by the spermatozoa and glycerol. It should contain fructose to supply energy to sperm and antibiotic prevent microbial growth. Extenders differ in composition depending on the species, use, temperature at which the diluted semen is to be stored, and the duration of storage desired. All extenders are based on a particular buffer, which has provided the best results for a given species. Hopkins and Evans, 1989. Buffers play an important role in modulating changes in the pH of extenders due to the products of metabolism in stored sperm. Carbohydrates, sugars, are necessary to provide the energy required for sperm glycolysis. Egg yolk-based fractions provide phospholipids necessary to promote membrane stabilization at lower temperature and limit premature acrosomal membrane activation. The commonest extender used for frozen semen is skimmed milk or homogenized milk to which 10% glycerol is added. Citric acid fructose glycerol yolk extender with 5% glycerol has been used with success. Merck, 1983. Packing of semen. Packing of semen is done in medium straw, 0.5 ml volume, and mini straw, 0.25 ml volume. Medium straw have a 3 mm diameter and mini straw have a 2 mm. They are pluged at one end, double plug end, with a sealing powder which is retained between two cotton plug. By applying vacuum to this end of the straw, semen can be drawn up the tube and into contact with the sealing powder. As soon as powder becomes wet, it turns into a gel to provide a very effective seal. Automatic straw filling machine use an ultrasonic pulse to seal the other end. Semen preservation. Semen is used either immediately after collection, fresh, for example turkeys, human beings, after storage at a reduced temperature, stored, for example horses, pigs, dogs, or after freezing and thawing, cryopreservation, for example, bulls. Cryopreservation. Semen is most useful for AI if it can be cryopreserved, since this method of preservation ideally enables the semen to be stored for an unlimited period without loss of quality until needed for AI. The spermatozoa are mixed with a protective solution containing lipoproteins, sugars and a cryoprotectant such as glycerol. These constituents help to preserve membrane integrity during the processes of cooling and rewarming. Straws are placed on rack and placed on vapors for freezing, and freezing is completed in liquid nitrogen. However, sperm motility must also be maintained, so that the thawed spermatozoa can reach the oocytes after insemination and fertilize them. In most species, the seminal plasma is removed by centrifugation before mixing with the cryo extender, for example, stallion, boar, goat and human semen. Thawing semen in warm water, 35 to 38 degrees Celsius, for 40 seconds is the most commonly used thawing procedure reported by inseminators, 80%. A significant increase in the conception rate, 27%, was reported when thawing occurred in warm water, 33 to 35 degrees Celsius, as opposed to air. Also reported a significant increase, 62.4%, in the fertility rate when thawing is done in warm water 35 degrees Celsius for 30 seconds compared to pocket thaw. Procedure for insemination. Firstly, it has to be ensured that the to-be bred is truly in heat. The restrained area should be familiar and free of stressful conditions. In the rectovaginal technique, a sterile, disposable catheter containing the thawed semen is inserted into the vagina and then guided into the cervix by means of a gloved hand in the rectum. The inseminating catheter is passed through the spiral folds of the cow's cervix into the uterus. The semen is deposited only 1/2 to 1/4 of an inch into the uterus. Slowly the plunger is depressed at the end where the right hand is, so that 1/2 of the straw's contents is deposited. Recheck and deposit the remaining 1/2. Timing of insemination. Correct timing of insemination is as important as correct placement of semen. Field experience has shown that the best results are obtained when the insemination is performed at or near the end of estrus. The beginning and end of estrus are very difficult to determine. The simplest practical method of timing inseminations is to use the AM/PM rule. Dual placement of semen. The reason that placement of semen both in the uterus and the cervix is recommended is that it helps to overcome the unpredictability of ovulation times. In cattle, ovulation can occur from 2 to 26 hours after the end of heat. Semen deposited in the body of the uterus reaches the fertilization site quickly to fertilize an ovum that is released earlier than normal. Semen deposited in the cervix survives longer. Due to its slow release from the cervix, it is more likely to fertilize an ovum released later than normal. Advantage: 1. Increased efficiency. 2. Improving animals' productivity. 3. Reduced disease transmission. Disadvantage: 1. Cost of AI compared to natural service. 2. Loss of genetic variation. 3. Difficulty of heat detection. Faults in AI technique. 1. Overexposure of semen. Frozen semen has a critical temperature of -70 degrees Celsius. If the temperature of the dose is raised above this level and refrozen, sperm will be damaged. 2. Incorrect site of semen deposition. Inability to pass an insemination gun through the cervix, or difficulty in doing so, is primarily due to inexperience. A greater and far more common mistake is to pass the gun too far into the uterus. This fault is prevalent, irrespective of experience, and is caused solely by inattention to detail. 3. Poor seal between straw and sheath. If the gun is loaded incorrectly, some semen will escape into the sheath and perhaps into the barrel. This reduces the number of sperm available for fertilization. 4. Neglecting to open the vulva. Opening the vulva before the introduction of the gun is a hygiene precaution of major importance. Irrespective of the cleaning precautions practiced, there will be heavy bacterial contamination of the vulva. Transfer of bacteria directly into the uterus is therefore inevitable unless the vulva is open.